Welcome, Immunogenicity 101. I have created these 9 statements as true or false. You can pause the video and you can attempt to answer them, or you can see how I'm going to be responding to these questions, answering them with the explanations during the talk. Immunogenicity is an idiosyncratic immune response to a therapeutic protein. So when a patient is injected a drug, their immune cells may perceive some of these drugs as danger signals. Now, an important concept here is that not all the time a drug is perceived by everybody as dangerous. The same drug could be perceived as perfectly normal by many people, and then a few people may react to this drug as dangerous. For some reason, the sentinel cells that we have circulating see something in the drug that triggers an alarm, makes them process this drug and present an epitope to the T cell with a co-stimulatory signal that makes this T cell differentiate into a T helper cell. This T helper cell is going to help macrophages, eosinophils, or neutrophils to attack and get rid of the drug. So this is a very specific response that the T cell is directing against that potential danger, because the antigen presenting cells presented an epitope that said, be careful, there's something wrong with this. Of course, the immune system does not know that it is a drug. It is just perceiving that there's something that's foreign and that could cause problem. So our immune system is triggered to make sure that we get rid of those danger signals. In addition to the T cell response, the T cell can also stimulate a B cell response, which is activating B cells, making them differentiate into plasma cells and producing anti-drug antibodies. In addition, the T cell can also stimulate a memory on these B cells so that they are ready always to get rid of the drug. So we have many different ways that the immune system will create an immunogenic response, and all of them are triggered to get rid of the drug because it's been perceived as a danger signal. Now, you would wonder, when do you want to have a response to the drug and getting rid of the drug? Well, sometimes we want this. Sometimes this has been tailored this way. And this is happening with vaccines. When you use the virus that's either killed or attenuated, or a piece of the pathogen, could be a bacteria or a virus, you can use a surface protein. You can use anything from a particular pathogen, and you make the immune system recognize this as foreign and trigger getting rid of the vaccine. And by doing this, you know that it's going to cross-react when the body encounters the pathogen whole. Our body is going to identify it thanks to the vaccine. Besides the vaccines, all the other drugs, we do not want this response. And also with the new generation vaccines that are based on nucleic acids, you don't really want an immunogenic response to the vaccine. You don't want your immune system to react against the genes. You want them to react against the gene products. And so in the case of nucleic acid vaccines and all other medications that we inject to patients, this is a not wanted response. And we need to be aware of it so that we can identify it and manage it correctly. When a drug is injected into the individual, and the drug could be intravenous, intradermal, epidermal, or subcutaneous, whatever way we inject them, the immune system cells that are traveling as sentinels, like the dendritic cells, can capture these when they identify a potential danger signal. They travel through the lymphatics, and in the lymph node, they present the T cell epitope to the T cell via the class II MHC molecule to the T cell receptor. This interaction, in addition with a co-stimulatory factor, will produce a differentiation to Th2 cells. These cells that have that particular T cell receptor and identify that epitope are now differentiated into Th2 cells, and they proliferate in the lymph node. also get activated and they express CXCR5, which is a migration protein. This brings the T cells from the T cell zone, which is in the center, brings them to the boundary where the B cells are. And when there's a B cell whose receptor also identifies the same epitope, this B cell will also express CCR7 that brings this B cell closer to the T cell zone. So the B cell is in the periphery, the T cell is in the center, but the CXCR5 brings these T cells close to the B cell zone, and the CCR7 brings the B cell close to the T cell zone, and bingo, this kind of interaction makes this B cell activate and differentiate into plasma cell. All of this activity and all of this energy is happening in the lymph node. So this is the reason why sometimes, if this is very profound and very quick, we may have lymphadenopathy or swollen and painful lymph nodes because all of this activity is happening in the lymph node. From the lymph node, the activated B cells will travel through the lymphatic systems, and in the subclavian vein, they drain into the blood and go into circulation. And the same thing happens with the differentiated plasma cell. It is the short-lived plasma cell, the one that is responsible for producing the antibodies that will bind the drug. And they will find it because now they are in circulation and they get to the site where the drug has been administered or where the drug is after absorption in the body. In addition, there's a special type of plasma cell that's called the long-lived plasma cell, and this cell will reside in the bone marrow and circulate through the blood vessels and go back to the bone marrow. They will be producing low level of antibodies to the drug. So when the drug is again seen in the body, these low level antibodies can help the body to get rid of the drug. Again, remember that the body believes the drug is a danger signal. So this is what they're doing. Depending on the sensitivity and specificity of the antibody assays, some of these persistent antibodies might not be detected. They may be so low titers that they may be read as negative. That is actually good news, because if you have a patient and you may have detected anti-drug antibodies, but then you don't detect them, it doesn't really matter if they have low levels of anti-drug antibodies. Bottom line, you can continue giving them the drug if they are benefiting from it, because they're so low levels that they're not really getting rid of the drug efficiently. Let's review the primary antibody response. And for now, let's forget the memory B cells. So when the drug is first administered and is first perceived as a danger signal by the naive B cell, this naive B cell will activate, and these activated B cells will differentiate into plasma cells. These are the short-lived plasma cells that will produce the first amount of antibodies. Now, to get here, remember that the dendritic cell had to first identify the danger signal, the drug. It had to travel through the lymphatics, go to the lymph nodes, present to the T cells, activate them. The T cells have to activate the B cells. The B cells have to differentiate into plasma cells and go back to the blood. And finally, we have these short-lived plasma cells in the system. This takes time. So after the patient has been injected with the drug and has been perceived for the first time as a signal, it can take anywhere between 5 and 15 days to have sufficient amount of antibodies in the blood, anti-drug antibodies in the blood, to be able to detect them with your assay. These antibodies are going to be the majority IgM. There may be a few that could be IgG, but mainly they're IgM. And as the drug is cleared from the system, this level of antibodies are going down. And this is basically driven by long-lived plasma cells which reside in the bone marrow, and they will be producing low levels of anti-drug antibodies. Let's take a look at the first questions. Immunogenicity is an unwanted immune response to any drug that is perceived as danger signal by the immune system. This is false because in the case of some vaccines, this is a wanted immune response, not in the nucleic acid technology of vaccines, but in all the others. Presentation of the T-cell epitope to the T-cell receptor in the presence of co-stimulatory signals triggers a Th2 differentiation needed for anti-drug antibodies. This is true. An adaptive immunogenic response is an attempt to get rid of a drug by the effector cells. We saw those are the macrophages and the neutrophils in the case of Th1 or Th17 phenotypes, and or by the presence of anti-drug antibodies, which is driven by the Th2s. Short-lived plasma cells are responsible for persistent anti-drug antibodies. This is not true. These are the long-lived plasma cells. In addition, in some cases, not all of them, there can be a memory B cell created, also triggered by T cell responses. And this memory B cell resides also in the bone marrow. Now, the fact that we have memory B cells is that they can proliferate and they can circulate also in the blood. And when they perceive the danger signal again, which is a drug again, they proliferate very quickly, producing very high levels of antibody. The advantage of this kind of response is that it's very quick, very fast against what is perceived as danger. So this is what we call the secondary antibody response, and it's mainly driven by memory B cells. So when the memory B cell has been created and there's a re-challenge of the drug being injected again, this B cell is very quickly producing plasma cells or differentiating into plasma cells that will produce extremely high levels of antibodies. This is what we call a booster response in terms of antibodies. The y-axis here has no units, but generally is in a logarithmic base 10 scale. And the titer boosters can be anywhere, 4, 5, even 30-fold or even higher fold compared to the first peak. And this is what is driven by the memory. They are mainly happening very quickly because these B cells, remember, they don't have to go back to the lymph node. They don't have to go back to proliferating T cells. They are already there in the bone marrow and they will produce these plasma cells very quickly. When the drug half-life is gone, the long-lived plasma cells will produce now baseline levels of persistent antibodies that are going to be higher than at the beginning. So the booster titer is the responsibility of the memory B cell producing very quick short-lived plasma cells in the secondary response, and the long-lived plasma cells after memory will produce persistent antibodies that are higher in titer than in the primary response. These are mainly IgG antibodies. Question 5. Long-lived plasma cells and memory B cells reside in the bone marrow and circulate to produce a booster ADA response to re-challenge. This is false because the booster is the responsibility basically of the memory B cell that will differentiate very quickly into short-lived plasma cells. The long-lived plasma cells in both cases, primary and secondary response, are responsible for persistent ADA. So now let's take a look at, we have the plasma cell differentiated and we have antibodies, but I did mention that in the primary response we have IgM, but in the secondary response they could have switched to IgG. In some cases, the switch can go to other kind of classes, including IgE antibodies or isotypes. Now, all of these isotypes identify the same drug. This is the same response, it's just that the class switch has occurred in the memory cell triggered by the TH differentiation. In the genome, we have the variable region for the antibodies that is responsible for identifying the drug. As I mentioned, all of these antibodies identify the same drug. So we will talk now about the switching and move to the right to the constant region. So what we see here is that the genes of the constant region go from left to right. You have a little circle that is identified with a capital S. That's the switching region of the genome. And then you have a rectangular piece in there that is identified with a capital C. That's the constant region. The Greek letters mu identify the IgM, delta IgD, gamma IgG, and epsilon IgE. And it goes in that order downstream in the genes. So what makes these cells switch is an interaction between that antigen presenting cell, that could be the B cell, into differentiated for a different Th or T helper cell that produces different kinds of cytokines and allows for a germline gene transcript that is an isotype switch. So for the Th phenotype differentiation, on the first antigen presentation of the epitope to the T cell with a particular costimulatory signal, the production of interleukin-12 or interferon gamma will differentiate the T cell into what we call the Th1 phenotype. It is characterized by the secretion of TNF-alpha, interleukin-2, interferon gamma, and interleukin-12. And the key target cell is a macrophage. The macrophages can deal with intracellular pathogens, tissue repair, or foreign bodies. And it was that piece of the first antigen presented, that linear epitope to the T cell, is the one that is shared by many of these intracellular pathogens or many of the different epitopes that will be presented when we have to deal with tissue repair or foreign bodies. So that's the reason why we get the Th1 response. All was driven by that antigen presentation. The mechanism by which the macrophages deal with these different responses is phagocytosis, killing, granuloma formation, for instance, with mycobacteria, and hypersensitivity type 4. And these can also be associated to an immunogenic response to the drug that has nothing to do with antibody production. It will be driven by macrophages. In a different antigen presentation and with different costimulatory factors, interleukin-4 will differentiate the Th into a Th2 phenotype with the production of interleukin-4, 5, 13, and 25. As you can see, the Th phenotypes produce cytokines that do not overlap. And these particular cytokines target eosinophils. The eosinophils are cells that deal with parasites and with allergens. And the mechanisms that they use to deal with these particular danger signals is production of antibodies. In circumstances of allergies, it's IgE, mainly what they produce. But they can also produce IgG or IgM, and they can fix and activate complement to get rid of these parasites and destroy them, kill them. They can also produce mast cell degranulation, and they are engaged or involved in hypersensitivities type 1, 2, and 3. Type 1 hypersensitivity is mediated by IgE. 2 and 3 have IgG and or IgM. And I encourage you to take a look at other videos that I have that explain those responses in more detail. In a different interaction, the production of interleukin-1, 6, 21, or 23 in addition with TGF-beta will differentiate this T cell into a Th17 cell with the production of interleukin-17, interleukin-21 or TNF-alpha, and those stimulate the neutrophils. The neutrophils will deal with extracellular pathogens like Candida, other fungi, bacteria, and the way that the neutrophils deal with these pathogens are by creating antimicrobial peptides, producing inflammation, maintaining the barrier integrity, but they're also involved in hypersensitivity type 4. So these three phenotypes are important, and all of them could be engaged in an immunogenic response to a drug. So there's not always involvement of anti-drug antibodies. So when we have anti-drug antibodies on a primary response, the plasma cell will be producing IgM. It is after that interaction that the TH phenotype may differentiate into different kinds of phenotypes with different cytokines in the milieu and influence the memory B cell by changing the germline gene and producing an isotype switch from IgM, and it can be all the way to IgE. In the human genome, the order of the genes is shown here. This is what I found from publications using human cells. If you're working or you are reading publications from any other kind of animal, not human, you may find a different order. But this is what I found for humans, and it always goes from left to right, starting with IgM and ending with IgE. So that particular interaction of the B cell and the T cell and the co-stimulatory factors will produce at the memory B cell germline gene the following. The two switching regions that I'm showing here, it's switching mu and switching epsilon, will join together, and all the intervening genes are going to be spliced out or deleted. So this memory B cell is going to have a new germline gene that only has a transcript for epsilon for the constant region of the IgE. So when it differentiates into a plasma cell, this plasma cell can produce only IgE. If you have a patient that has an anti-drug antibody and it's an IgE isotype, and let's say it presented with an anaphylactic reaction characterized by IgE, that particular individual will always have all of the anti-drug antibodies that are going to be IgE, because that patient basically has spliced out all the genes for all the other constant regions, right? Wrong. This B cell response, as we remember from the lymph node and this proliferation, is it's driven by a polyclonal response. The B cells can do somatic mutations, and they can actually find several different variable regions and mutate to bind the drug better, but there's different cell lines, there's different clones. So it's a polyclonal response, and it's also polyclonal at the level of the plasma cell. So remember, some of these plasma cells can still produce IgM. Some others may have switched to IgG. Some others could have IgA, which is not very common for anti-drug antibodies. Some others may have IgE. So bottom line is that you may have an individual that could have had an IgE response, but that individual could also have other responses. IgE could eventually wean down and disappear, but the patient could maintain having anti-drug antibodies of the IgM class because it's a polyclonal response, but all of them will identify the drug. So that's why it is more important to understand if you suspect anti-drug antibodies. Don't worry about the isotype, look at the levels or titers and look at the assay for anti-drug antibodies. That should be specific for the drug that you are using. In terms of driving these Ig isotypes or these class switches, this is another table that is challenging to put together, and my recommendation is use this as an example, but you can read more papers and you can build your own table and have a better idea if you need to work on this area. And the reason why I'm saying this is you need to read very carefully between the lines, or I should better say in the method section. Some of these are using one kind of cell line, another might be using a different kind of cell line. The cytokines they're using might be from different vendors, different affinities, and bottom line, the conditions of the assays are not the same for all the papers. However, what I was able to do is a more comprehensive way of saying, in general, what I found is that the Th2 response driven, basically driving IL-4 cytokine production, will permit a switch to IgG1, IgG2, IgG4, and IgE. Now, we actually care about IgE because this can be an anaphylactic reaction, which we all know could be life-threatening. So it's the only one we care about, right? Well, wrong. We care about all of them because some of the IgG can fix complement and it can cause necrosis. You may have anti-drug antibodies that if the mechanism of action binds a surface, let's say a surface antigen on the kidney, and you get anti-drug antibodies of IgG and now they're binding kidney, you can have serious kidney disease. Driven by these anti-drug antibodies, and it's not mediated by IgE. So you need to understand the mechanism of action of the drug and the potential for immunogenicity and understand what you are reading if you are getting results in terms of anti-drug antibodies, but also in terms of predicting if you work on the drug development area. You need to make sure that you understand how to predict what is the more likely response. Interleukin-5 will stimulate switching to IgA, interferon gamma to IgG3, TGF-beta to IgA, and interleukin-10 to IgG1 and IgG3. In addition, I found that IL-4 decreases the likelihood of switching to IgG3, and interferon gamma decreases IgG1 and IgE switch. TGF-beta decreases the switching to IgG3. So what do you need to take into account when you are considering a drug that you're giving to a patient or when you are in the drug development area? This is more useful for identifying potential risks for drug development. Well, the first thing you need to remember is you need to identify, is there any known T-cell epitope in the drug that could be recognized and could provide a stimulation to any one of these Th phenotypes. Evidently, in drug development, when this particular epitope is identified, all the efforts are being made to change that molecule, get rid of that epitope, and make sure that the drug still works. Now, sometimes that is not possible. Sometimes that epitope needs to stay in the drug for the drug to work. And so we do know that there's an increased risk of immunogenic response. On the other hand, you need to look at the mechanism of action. So let's say that you have a drug that has a potential epitope that could trigger a Th2 response, and you cannot get rid of it. And this drug, the mechanism of action is inhibition of interferon gamma. So now you have, on one hand, an epitope in your drug that may increase the amount of IL-4, but the mechanism of action of the drug is to decrease interferon gamma. So now you have a higher risk of probably getting anti-drug antibodies of the IgE class. So what you need to build in the drug development program is a careful examination of potential anaphylactic responses by looking at Samson criteria and by carefully detecting, collecting, and reporting all of the adverse events. And of course, making sure that when a patient is given the drug, the patient is observed for at least 1 hour after the injection so that you know if there is an anaphylactic response or not. If you're not doing drug development and you suspect that the drug may have this potential risk, just read the package insert or the label that you have for that particular drug that you are prescribing to your patient so that you understand what is the kind of risk that you need to follow. The differentiated Th phenotype drives the specific isotype switch to produce anti-drug antibodies of IgM, IgG, IgA, or IgE isotypes. That's true. Anti-drug antibodies can switch isotypes in any individual at any time after administration of the drug, and it is idiosyncratic. This is true. The mechanism of action is the key to predict potential isotype switch of anti-drug antibodies. That's not true. The T-cell epitopes are going to be critical. While the mechanism of action can produce an increased risk for IgE if the drug does not have an epitope that identifies the drug as danger. This individual may have an IgE response, but not to the drug. T-cell epitopes and mechanism of action are considered to de-risk and assess the impact of immunogenicity of the drugs, and this is true. Immunogenicity in a nutshell. So when a drug is perceived as dangerous by the antigen-presenting cells and there is Th stimulation, the Th can differentiate into a Th1 that will stimulate the macrophage, Th2 that targets the eosinophils, or Th17 that targets the neutrophils. And all of these are going to try to get rid of the drug because the drug has an epitope that has been identified as a danger signal. And this can have evident effect on efficacy and or safety. So if these cells are trying to get rid of the drug, you may have less efficacy than if you don't have an immunogenic response. On the other hand, this immunogenic response may not be very good at getting rid of the drug, so it could still be efficacious, but it may not be safe because you may be having inflammatory reactions. You may be having some manifestations of adverse events. Now, on the other hand, you may also have these cells may not be dealing with the drug very efficiently, and they may not be causing any safety effect. So immunogenicity needs to be assessed as a whole. Is it causing an effect on efficacy and or safety? On the other hand, we have the B cell response, and this plasma cell will produce anti-drug antibodies, same as if you do create memory, or if the individuals create memory to this drug, they will be producing booster responses to the drug. All of these can also have an effect on efficacy and or safety. Now, because we have here a test that can be measured in terms of anti-drug antibody titers, some of the pharmaceutical companies, when they develop the drugs, are producing modeling of pharmacokinetics or pharmacodynamic against the titers of the anti-drug antibodies. And this will be surrogates of efficacy. So if you can see in the package insert or in the publications that there was a direct effect on titers of antibodies and decrease of pharmacokinetics that would predict loss of efficacy long term, these drugs oftentimes are not further developed because they may fail long term to produce efficacy. In other cases, these drugs may actually be developed because the likelihood or the risk or the percentage of patients that produce these high-level antibodies is very, very low. So you may need to put all of this in context when you read the package insert and when you read the label. If you are developing drugs, of course, this is something that you need to do because it's a regulatory requirement to have a complete and comprehensive assessment of immunogenicity when you develop the drugs. There were several publications that I used for creating all of this talk, and I recommend you to continue looking at more recent papers because this is a very active area of research, and also take a look at my YouTube channel for other educational videos. Consider subscribing, and I hope that you enjoyed this talk. Thank you so much for watching this video, and stay healthy, happy, and well.